Okay guys, so here I am drawing the structure of a gastric gland from anywhere from the fundus or from the body of the stomach. This is a gastric gland. Here we are having the mucus layer. This is the mucus layer. Now the surface cells are here. These are the surface cells. They will produce mucus, trefoil peptides along with the bicarbonate ion secretion. This is actually the epithelium between the glands, because here we are having another gland this way. Here we are having mucus neck cells. These are the cells in the neck. Now, what is the function of these neck cells? They act as stem cells. Further, here we have the parietal cells. These are the parietal cells. They will give us HCl acid and intrinsic factor. Now further, here we are having this ECL cells. This ECL actually means enterochromaffin like cells. Enterochromaffin like cells. They are for the production of histamine. Now at last we are having the chief cells. At last we are having these chief cells for pepsinogen secretion, these zymogen cells for pepsinogen secretion, plus this gastric lipase will also come from these chief cells. Lipase will be using for the digestion of the lipids. Now these gastric secretions are arising from these glands that are in the walls of stomach, most probably in the fundus in the body of the stomach. This mucus layer is being formed by this mucus, bicarbonate and trefoil peptides. This trefoil peptides will stabilize this mucus layer, and this is because so that the stomach cannot digest itself, because the environment is so acidic. Now this acid that is being produced by these parietal cells, this acid will sterilize your meal. This acid will sterilize the meal, and it also causes the hydrolysis of dietary macromolecules. In your diet, all the macromolecules, first they are being partially digested by chewing, the process of mastication. Further, these macromolecules will be hydrolyzed by this acid. Now, what is the function of this intrinsic factor? This intrinsic factor is specifically for the absorption of vitamin B12, and the other name for vitamin B12 is cobalamin. This pepsinogen we have discussed earlier will be acted upon by HCl to convert into its active form, the pepsin, and this pepsin will be used for protein digestion. And I told you before that this lipase will be further used for lipid digestion. Now, what are the factors that are the stimulus for these gastric secretions? Number one, we have a hormone called gastrin. This is a hormone that is coming from the antrum of the stomach. Here is your esophagus, here is your stomach, and here is the duodenum. Now, this is the antral region of the stomach. This is the antrum. In the antrum, we are having these G cells, and from these G cells, we are having this gastrin. Now, what are the factors that release this gastrin from the G cells? It can be a response to GRP. It can be a response to GRP, a gastrin releasing peptide, also known as bombacin. It is a neurotransmitter, and this because it is a neurotransmitter, it will ultimately be coming from the nerve endings, and the nerve endings from where this GRP is coming is the enteric nerve endings, the enteric nervous system in our stomach, all over the GIT. Enteric nerve endings giving GRP, and this GRP is stimulating the release of this gastrin. This gastrin can also be released from the G cells of the antral region of stomach in response to the presence of oligopeptides, that is partially digested peptides, in the lumen of our stomach. Because this is triggering, this is giving the message that the peptides are not yet being digested. So it will ultimately be giving a positive trigger to release of gastrin. And through the bloodstream, the gastrin is carried to the fundic glands, the glands in the fundus, here, in the fundus of the stomach. And here, this is the gastric gland. This I have drawn is the gastric gland. This gastrin will go and bind on these parietal cells, these chief cells, these ECL cells. All these three cells, parietal, ECL, chief, all these three cells are being bound by gastrin now. Now what will happen next? All the secretions of these three glands will be stimulated. One more thing, this parietal and this chief cells can also be stimulated by acetylcholine, can also have a positive effect on parietal cells and these chief cells. So ECL cells can be stimulated by gastrin. Parietal can be stimulated both by the acetylcholine and gastrin. Both are this is a hormone, this is a neurotransmitter. And in the chief cells, same as parietal cells, can be stimulated by this acetylcholine and can be stimulated by the bounding of this gastrin into this.